Nice to see you.
Where are you, Javier?
At Twente, at the University of Twente. So I'm
nice.
on a sabbatical here, invited by Detlef, Devaras, and others in the group. Here is Detlef coming to from the other cameras.
Hello, Detlef.
So he fortunately is here. So we are enjoying very much that he is here. So
yeah.
see, I only go to next door. So
yeah, exactly. So it's as easy as crossing the hallway.
Very good, very good. Very good.
I'm looking forward to the talk. So
yeah. Okay. So Javier, please start showing your.
Sure. I will start sharing my screen. Let's see how. Oh, I'm going to share the whole screen, right? Screen one.
Yes.
Okay. I think now you can see the slides.
Yeah, now it's full screen. Yes, perfect. Okay.
Perfect.
Okay. Then, yeah, just a few seconds and we'll start. So we have a good number of participants. All right, so just. Okay.
No biologists in the audience, no?
what?
Sorry?
No
biologists
in the audience, no?
From
yeah, it was a special story. I tried to invite people from bio or even from other schools here, but then I found that actually they receive information about each our seminar, but so far nobody attended. So that is the amazing. So very special, situation with mathematics. Yeah. Okay. So we are starting, right? So I'm happy to welcome today Javier Rodríguez. He is one of the best friends of UEA for many years. I believe the contacts between Javier and UEA started with Norman Riley, and then Javier worked and cooperated with Mark Bly, with other people in our school. I was involved in several papers done by Javier, so it was a very successful cooperation. And you could remember his presentation in person before pandemic on beer and bubbles in beer and 2 bottles, and if you impact these bottles in special way, so you can cause the cavitation. And today presentation by Javier, it will be kind of related to biology and could be pandemic, could be to COVID situation, I believe, because it's at least the first slide reminds me the mask and how that small droplet goes through the mask. It could be related. But I not familiar what will be inside, so I'm waiting the presentation the same way as all other participants. The Javier was kind to allow us to record his presentation, and it will be published in YouTube. And it's very interesting that our videos, our seminar, receive the quite good number of watching. How to say this? But I was surprised that the champion in this field is Stefan Zaleski. Who already his video was watched almost 400 times.
Wow.
And I checked he's.
Sasha?
But it was indeed.
Sasha? I'm missing you. Hello?
So, yeah, you are fine. I think Sasha has a problem, but you are fine, I think.
Okay, thank you, Detlef. Yeah, I'm missing Sasha.
You should just start. So Sasha may be able to hear you, but we don't hear him. He is frozen. So,
is there anyone else from UEA who can copy? Copy, like hear me. Sorry, I'm using the radio.
Yeah, we can hear you. Can you hear us?
Hi, Javier. Yeah, we can hear you.
Javier, we can hear you.
Yeah, we are here in the tea room. Lots of us from the School of Mathematics, and everything works fine. I think we just lost Sasha.
Okay. Oh, but
Javier, sorry. I still have problems with the computers. Can you see me now, Javier?
Well, I cannot see you because my screen is now on presentation mode, but I can hear you.
We see you, Sasha. We see you. You are not
Okay. I'm very sorry. So the two computers and they crash one after another. Then Javier, please start your presentation. I'm very sorry about internet. Yeah.
Computers.
Yeah. And please, may I ask all participants to mute their microphones and videos? Then quality will be a little bit better. Okay. Okay. Yeah. So then Javier, please start. I'll check with participants that all of them are muted. Yeah, please start.
Thank you very much. Well, first of all, I'd like to thank Sasha and the fellows at the School of Mathematics at UEA for this very nice invitation. As Sasha said, the history of my collaboration with UEA starts to be long, and it started many years ago. And it's always a pleasure to be in contact with you guys. We haven't seen each other physically for a while due to, well, for obvious reasons, but I hope that I'll be able to visit you in person soon this spring, hopefully. Actually, when Sasha mentioned that some of you are in the tea room, it brought me very good memories of coffees and lovely scientific discussions in that room. So I hope to be able to have them again with you guys soon. Well, so today I'm going to talk about something that is different to other talks that are given there at UEA, online or physical. Actually, today's, it's something that I'm working as a side project with some colleagues from the bioengineering department of my university. And basically, we want to understand how is the interaction of a biological tissue, of a cell tissue, with a flexible object. Sasha mentioned at the beginning that the applications are, well, certainly very different to the ones of the beer bottle project that I had. They are not related to COVID. But doesn't have anything to do with the virus, although it has to do with some hopefully relevant applications in biology. And he was mentioning about the image on the cover. I don't know, does anyone want to guess what this image is? No?
It's fiber, which.
Exactly.
And the kind of droplet, or what's that?
Cells. It's a skin cell. It's a skin cell. Yeah. So this is the. Today's topic is going to go about the interaction of cells with flexible objects, and indeed, what you see here on the cover is a skin cell, what is called a keratinocyte. It's a skin cell that is grabbing on a carbon fiber to try to move it. Actually, I must confess that scientifically, this image is not very relevant. It's mostly aesthetical, and actually is the one of the co-authors of the study that I'm going to describe today. She did her PhD thesis on this topic, and she wanted to have a nice image for the cover. So basically, she took this image with the electron microscope, with SEM. The scan electron microscope, to have like a detailed image of how a cell physically grabs a carbon fiber and supposedly pushes it. However, it's very unlikely that a single cell is able to do anything relevant with this fiber because it takes a lot of them. It takes a tissue. So that's what I say, that the image is super nice. I love it. It was the cover of her thesis, as I said, but it's not exactly what I'm going to talk about today. I'm going to talk about cell tissues when there is a collective motion of cells. In fact, the motivation that we have is, well, we want to understand some aspects on the collective cell migration. Why collective cell migration? Because it's a problem, a biophysical problem that appears in plenty of very relevant biological or medical situations. Perhaps one of the most famous ones, or at least one of the contexts in which it has been studied the most, is in the context of wound healing. Wound healing is the process by which a tissue that is meant to be continuous, let's say, think for instance of the skin, suffers a wound, and then the cells that surround the wound suddenly start to coordinate with each other, and eventually they orchestrate a coordinated response that eventually closes this wound. We have the first video, the video that you see on the top right of the screen, is actually an experiment that we did in the lab many years ago, where we have what is called an epithelium of skin cells. So basically, it's a monolayer. It's a layer where there is only one cell in depth, and we produce artificially with a tool. We produce a circular wound of about 1 millimeter. Of course, this kind of wounds are very small compared to realistic wounds, but it's nice because you can get to see how cells coordinate to close this wound. Basically, the reaction of cells is twofolded. They do two things to close the wound. On short time scales, what they do is to migrate, as you can probably see on the video. So they basically start to walk towards the center. However, at longer times, and especially when the wound is kind of big, definitely much bigger than this 1 millimeter circle that you can see on the video, basically just by migrating to close the wound is not enough. You can easily imagine that a cell can only stretch so much. I mean, it cannot multiply its size by a factor of 10. So at some point, if it needs to cover a very large wound, then of course they need to replicate. They need to divide, and they need to proliferate and to create new cells. But let's say that still, even if the process is complicated from the biological point of view, there is a first stage in the process where the cells are just migrating, walking, if you want, or crawling, we should say, towards the center. So this is one of the most relevant biological situations where cell migration is important. The other one is what is called morphogenesis or embryogenesis. To present that, I have another video that we didn't do in our lab. Is taken from this video that is shown there on the bottom right corner. So basically, morphogenesis is not playing. Let me see if I can play it again. I was too brave. Yeah. I think it's playing again. Yeah, you can see that. So that's another situation where collective cell migration becomes important, which is in morphogenesis, which is basically the development of tissues and organs. You know that an organism always starts from pluripotent cells that stem cells that can differentiate into any organ, any tissue that is needed. So what happens many times is that this cell differentiation process takes place in parts on the embryo that are very far away from the final destination that this organ will have. So what happens sometimes, as you can see here, especially at the beginning of this video, is that cells differentiate into a tissue, and then they start to migrate collectively when there are already enough of them that have differentiated to occupy the space where eventually the organ is going to be developed. So of course, without cell migration in the process, morphogenesis wouldn't be healthy, wouldn't take place as we know it. So therefore, it's a super relevant problem. And the last one that I want to mention, although I have no material to show, but I think we all have it in mind, is metastasis. In metastasis, what happens is that tumor cells migrate collectively to invade healthy tissue. And at some point, these cells actually split, and individual cells are able to detach from the main tissue and go on their own, like scouters. But basically, there is an initial stage in which cells migrate collectively. Well, this is very broad, of course. So I'm going to talk today about only one kind of migration, which is the migration of epithelia when they are like the layer that you see in the first video, when they try to close a wound. The main motivation is because the people that I work with at my university, the people in the bioengineering department group, they are very applied biologists. Actually, they work in hospitals, and what they do is to produce artificial skin. So they produce this artificial skin for a variety of diseases. Well, the most famous one is to treat people with a high percentage of their bodies burned because of an accident. So they are able to produce or to manufacture, if you want, skin to be able to repair the natural skin of these patients. But also they work with people that have rare disease that are most commonly caused by mutations, and that for one or reasons that are not always well understood, these mutations preclude the skin of these patients to close wounds or even to have like normal functions, like for instance, remain uniform and remain how do you say, continuous with no breaches, no? So these people work on this kind of problems, and you know, basically they want to understand what happens in some of these diseases, why some skins are not able to grow normally, or some others they start to have like blisters all of a sudden, and other things that happen to these patients. And one of the hypotheses that they started to work on many years ago is that mechanical forces are very important to regulate cell migration and wound healing in this kind of disease. So what they speculate is that some tissues are not able to exert enough force on the surrounding tissues, and these surrounding tissues actually need need this force to be able to trigger or to be able to regulate its cell migration. This is also well known that this happens in many cases. For instance, tumor cells, they are very well known to be affected by the stiffness of the surroundings. So when they are found in a stiff environment, they are more able, or they are better at migrating, right? So this is well known that this happens with some kind of cells. But it's not clear what happens with some of the mutations that my colleagues study, especially because, as I said, they deal with rare disease. So we are talking about disease that only a very small percentage of the population have. So they don't really have a lot of evidence, and they have a hypothesis that they need to try. So the idea that they had in mind was basically to be able to measure the force that a tissue is able to exert as it migrates, something as apparently simple as that, no? Of course, there are many techniques that have been put in practice over the years in biology, but these techniques actually are not very useful for the kind of things that they do. Well, because sometimes these techniques require like complex microscopic tools or microscopy tools to be able to be implemented, or because they don't give the kind of information that they are interested on. So, I mean, basically they want to be able to measure the force that their tissues are able to exert on the surroundings and how these forces affect their migration. So that's why we started collaborating, trying to design a technique that can be validated in 2D, because most of the experimental techniques that people can use to study wound healing or cell migration are 2D, but also a technique that could be as simple as possible to implement in 3D, and in a way that could actually be practical for the clinic. So something that can be used to to understand the role of forces in the progression of this disease. And well, this is the interest they had. Of course, I mean, I have the same interest, but also as a physicist or engineer, I also want to understand the biophysics behind the interaction of these cells with the forces that surround them. So we also want to do some mathematical modeling of the process, and try to see what this mathematical modeling is able to explain about the mechanics of these cells, no? So basically, this is the motivation. You see, I mean, it's a very applied problem, although I'll try to do some nice mathematics with it and something more fundamental. Basically, yeah, we want to measure the forces that these tissues exert as they migrate, and to do some physics to understand this process, no? So the idea that we had some years ago, it was in principle simple. It was, let's have an epithelium that is migrating to close a wound, and then let's put some obstacle in the way. So let's put some flexible obstacle that the cells are able to deform. So, well, the experiments that we did is what is called the wound healing assay that is very much used in cell mechanics. Basically, the biologists grow a continuum infinite or very large cell tissue, and then at time 0, when the experiment starts, what they do is to remove part of the tissue. So what happens is that the cells that are here at the front of the wound, then they realize quickly that, how the tissue is gone, and they start to orchestrate this response to close the wound. What we do is to set a little trap to them. What we do is to put this fiber, which is actually a carbon fiber, like the kind of fiber that is used in composite materials. So we place this carbon fiber on the way. So you are going to see that these cells migrate, and they, meet the fiber and start doing things with them. So with it, I mean. So this is interesting because, of course, when you see how a fiber deforms, the mechanics of the fiber is very well characterized since the 19th century or the 18th century. So the deformation of the fiber is going to be used or we are going to be able to use it to measure the force that actually the cells are exerting. Of course, I'm going to show a video of this very nice experiment. So this is what we see in our lab. So you see that this we have the cell monolayer. The migration, as you see, goes from top to down. What you see here, this red line is the carbon fiber. Of course, the carbon fiber is not read in this microscopy technique. This is the image detection that we do by computer. And I show the image detection because otherwise the fiber is kind of hard to see, right? So you see what happened. As expected, the cells, when they get to the fiber, they start to deform it. And what happens that at the beginning, they start to move right like if there was no fiber. So they keep moving with the same speed that they had when they were closing the wound. But then at some point, they stop. They stop kind of all in a sudden.
Javier, may I ask you, the scale, time scale, is it in hours?
It's in hours. I think that, unfortunately, the quality I don't think is very good. But yeah, it says it's in hours. The numbers that you see on the the numbers, these are very long experiments. So yeah, all the videos that I'm going to show today, they are very they are sped up a lot.
Okay, thank you.
Thanks to you. Yeah, so it's it takes several hours for cells to move of the order of the size of the fiber. And the spatial scale, for you to have an idea, this fiber is probably about 3-4 millimeters long. So this is the scale of what we are seeing. Anyway, so this is what we see that happens in the experiment. And of course, as I said, the first thing we wanted to do was to use this deformation to measure forces. So what can we say about the motion of this fiber? So, well, the first thing we tried, and actually it worked, it was, we have time lapse, we have videos, so we are able to see the deformation of the fiber at different times, see the time scale of the experiments. We are talking about days, like 5 days, something like this. So what we do is to get the experimental shape that we obtain from the videos. This is shown in blue in this plot. And then we compare that with, would you expect a beam to do in classic rod theory or beam theory? So the first beam that we tried is the simplest one, which is Euler's beam, which is an elastic beam of constant properties or constant cross-section and working in the small displacement regime. And to our surprise, as you can see, this equation fits very nicely the experiments. I say to our surprise because if you see the deformations that we have on the right, they don't really look small to me. So I use them there to linearize the equation and to say that this is linear displacement. But it happens that, as you see, this equation fits very nicely the experimental shape that we measure from the experiments. That's nice because it means that we can then use the equation that you see on the screen, the equation for Euler's beam, to find out by matching the deformation with the equation to find out the only unknown that we have here, which is F naught. F naught is the uniform forms, the forms per unit length that is applied by the tissue on the beam. And you see, as I said, that the agreement is reasonably good. So basically, we can use this technique to measure the evolution of the force as a function of the time. And this is what we did for in different conditions. In particular, the main parameter that we change is the length of the fiber. And we observe a number of very interesting features. The first one, as I said, is that initially cells move at a nearly constant speed, which is, well, at the beginning, you are not surprised because at the beginning, the fiber doesn't really oppose much resistance, right? But then at some point, and here the deformation is actually quite large towards this part of the curve. At some point, what happens is that the fiber is strongly deformed, and presumably it's already exerting an important force on the cells. And still, you see that these cells keep moving at a pretty much constant speed. So that's something that the model that we will put together later on will have to explain. Why actually we see this constant speed, no? Even though you are exerting already a force on the cells. So that's one thing. The other feature that we observed is that in most experiments, in particular in the experiments that correspond to the shorter fibers, here as you can see on the color scale, blue is shorter and yellow is longer. So in the experiments that you see, especially at shorter fibers, what happens is that there is a maximum force. And after the system have reached this maximum displacement or maximum deformation, what happens is that the cells are not able to keep up with this force. They are not able to counteract the force of the fiber, and they start to recoil. The recoil is very apparent, as you see on the short fibers, but it's not so apparent on the long fibers. On the long fibers, what happens to see is that they reach this point of maximum deformation, and they keep it. They are able to stay there for as long as we can measure, no? Which is about 5 days. So this is the other interesting effect. Something else that we observe in this experiment is that the force happens to be stronger for the shorter fibers, which is kind of counterintuitive if you think about it. Like if I'm able to do a given force, then, well, my force is independent on the opposition that I suffer. It's like if I have something that is stiffer, then I do a stronger force. This is not something that is very intuitive, but it seems to be the case here. It seems to be the thing that cells are doing. And by the way, this is something that you also see in other biological systems. For instance, as I said before, in metastasis, typically tumor cells respond doing stronger forces or migrating more when the environment in which they are is stiffer. So actually, it seems that our cells are doing kind of the same, no? are having kind of the same behavior. Well, so here's something that I think it's clear, but I like to put the equation. When we talk about a longer fiber, we also mean a stiffer fiber. In fact, if you take the equation that I showed before, the equation for Euler's beam, if you calculate an equivalent stiffness, which you can do, for instance, differentiating the force with respect to some characteristic deformation, say the one at the tip, then you get that the stiffness of the fiber, the elasticity constant of the fiber goes as the product of Young modulus times the moment of inertia of the section divided by the length of the fiber raised to the fourth, which means that indeed, as common intuition says, long fibers are typically easier to deform, are softer from that point of view. So these are the things that we see in our experiments. And then, well, as I said, we would like to do some model on this, some mathematical model. And of course, if you want to model the whole motion of the tissue, that's very complicated because there are plenty of effects. And actually, there are already models in the literature, plenty of them, to describe how a tissue is moving, free of any influence. So what we wanted to do in this work is just to describe with the minimum number of elements possible, and that's something that is going to be a guideline in this study. We want to describe with the minimum number of elements possible the interaction of the cell with the fiber. So we are not so much worried about what happens far away from the fiber, far away in the tissue. We want to describe the main features of this phenomenon. And for that, of course, we need to look at experiments because this kind of systems is too complicated to do theory without looking at what cells do in the lab. No, so these are the different ingredients that, these are the in different colors. I marked some of the main observations, and here I'm going to show some. Sorry, yeah, I think I'm hearing myself. I don't know if someone.
Yeah. It's sorry. It's a new participants arrived and didn't mute his microphone. So okay. now he is muted. Yes, and now it should be okay.
Okay, thank you very much. Yes, as I was saying, sorry, I was a little bit disturbed by into myself. So yeah, so here in different colors we have some of the main observations that we do from the experiment. And on the right column, I'm going to show which ingredient we want our model to incorporate to be able to take into account that effect. So the first thing is that we see that cells move initially at a constant speed and then at some point recoil. It's true in some cases they stay with a constant deformation, but typically when the fiber is very stiff, as you saw, they actually recoil, right? So this is a behavior that reminds us very much of a damped oscillator. And in fact, to be able to obtain such a recoil, you need some inertia in your system. If you only have viscosity or dissipation in more general terms, then what you see is that you asymptotically approach the limit, but you then expect a recoil. So a recoil basically means that we need inertia in our system. So we are going to incorporate some inertia-like features. You will see why I don't dare to call it inertia. I rather call it inertia-like feature. So we have this inertia. On the other hand, of course, well, we see that the oscillations don't last forever. As a matter of fact, we only have a single recoil, and in some cases, you approach, as I said, asymptotically to the limit, to the maximum deformation, which means that we also expect to have some viscous-like dissipation, some sort of mechanism that is able to dissipate the energy of the cell tissue. The other thing that we need, for sure, the third ingredient, is going to be the elastic force. So we are going to need some elasticity, some elastic element in the system that will replicate the effect that the fiber has. And this is a hypothesis that we are going to do, and then we'll discuss about it. But we are going to assume, for sake of simplicity, that the fiber stays always at the front of the tissue. You probably remember from the video that this is not really the case, that some cells are able to jump over or pass below. I think they actually do the two things and pass the obstacle. But let me use this assumption, okay? So this is basically the ingredients we want to have in our model, no? So far, so good, because I'm describing elements that you can place in any continuum model theory. However, when it comes to cell biology or to biophysics at the cellular or tissue level, inertia is kind of a problem, right? Let me show you why. Well, first of all, inertia is there. So I explain our problem. I explain that our cells are able to reach a maximum and then recoil. That's a very inertia-like phenomenon. But I mean, this kind of inertia-like effects are everywhere in tissue mechanics. So what you see here is another experiment that we did in our lab. This is another of these wound healing setups where we have a continuum layer and we produce the wound. And this time, the wound, as you see, is kind of narrow. It's about 1 millimeter, but instead of being a circle, like in the first experiment I showed you, now it's going to be a band. Okay, so we have these two populations of cells that are confronting each other. Of course, what they want to do is to close the wound, right? But then look at what happened once the wound is closed. So you see that, yeah, they come together. Of course, when they come together, the tissue is closed, and you expect that the stimulus that makes cells move stops, because, yeah, there is no wound anymore. And still you see that cells, you see, keep their motion. So they keep migrating, and they do it in a fashion that remembers very much when you have like two water waves confronting each other on a beach or something like this. No, you see that this is some sort of wave. Yeah. So clearly there is a lot of inertia in this movement. No. However, as I said, inertia is not easy to account for at these very small scales, and you can imagine why. Basically, well, if you calculate a Reynolds number of this flow, what you have is, well, the density is typically the density of water, because cells have pretty much the density of water, and they are surrounded by water, of course. The typical velocities that we find are of the order of 1 cell size per hour or per several minutes, so let's say 10 microns per hour. And the typical size of the tissue, at least in this kind of experiments, is 1 millimeter. Now, if you divide that by the viscosity of the liquid where they move, which is like the one of water, then what you get is that the Reynolds number is extremely small. It's about 10 to the minus 6. So it's quite remarkable that you can see this inertia-like behavior when physically there is no inertia in the system. Now, what is going on? What is then causing this what we call inertia-like features? Well, it's something that happens in cells that is called cell polarization. Polarization basically means that inside the cell, when the cell gets the order that it has to migrate or decides based on some biochemical clue that it has to migrate, what happens is that there are gradients of biochemical molecules or biomolecules inside the cell. Like here, for instance, this is an image that we took from a recent paper. Here you have an amoeba, which is kind of a big cell. And this amoeba, you can see that it has different molecules on the front, which is the red part on the right, and on the back, which is the green part on the back. On the front, there is what they have is actin, which is one of the elements that form the cell cytoskeleton. So you see that when the cell migrates, start to build new cytoskeleton, and this is the red part that you see at the front. And at the back, there is a contractile molecule that is called myosin, that basically once the cytoskeleton is formed, what it does is it's sort of like a tendon. It tries to get the fibers of the cytoskeleton together to increase locally the pressure and create a flow of the cell to the front. So this is the kind of thing that happens when a cell is moving. Of course, to induce this grading of biomolecules, it takes time. So it's not something that occurs instantaneously. Typically, it takes several minutes for this gradient to establish. And conversely, you can imagine this gradient takes also minutes to disappear because, basically the cell keeps a memory of what is the state of motion through this gradient of molecules. And therefore, this state of motion, once it's set, doesn't disappear instantaneously. It decays with time. No? So this is basically the effect that is behind our inertia. So it's not really inertia because, of course, the velocities and the size and everything is just too small. But mathematically, and this is the point that I wanted to make today, mathematically, we are going to model that effect as if it was an inertia, which, by the way, for people coming from fluid mechanics is extremely comfortable because we have plenty of ways to deal with inertia in our equations, and it's something that is kind of pleasant for us. So this is, as I said, this is a picture that I took from a paper, because in our experiments, we also tried to look for polarization, like you are seeing here. But it proved to be harder. Actually, amoebas are cells that keep an excellent polarization and they move super fast. So if I could pick the cell to show polarization in, I would pick an amoeba. But we don't have amoebas, we have skin cells. So when you do the equivalent experiment with the skin cells, things are not so clear. So that's what I borrowed this image from an article. No? So here what you see is a fluorescent microscope image of the cells surrounding the fiber. You cannot see the fiber very well on the blue image because, you only see the nuclei. The blue spots are the cell nuclei. But if you go to the other image, to the red one, there you see the fiber very clearly. And you see this accumulation of like red spots or red pattern in the tissue. This is the same red molecule that you saw on the previous image, this F-actin. So yeah, ideally we would like to see the polarization in our cells. However, it's a lot harder because what happens is that our cells are not isolated. Our cells are in a tissue, so one sort of sits on top of the other sometimes and grab on each other, or I mean they do funny things, and therefore it's not so easy to see the polarization. Yeah, but the same molecules are there, as you can see on the image. So yeah, of course we expect our cells to have a strong polarization and therefore to be able to reproduce some inertia-like features. Well, so this is basically the other one of the effects that I was mentioning. The other thing that I wanted to discuss briefly about before going into the mathematical details of the model is putting the fiber at the front. So actually, as you saw in the different experiments, many cells are able to go past the fiber, right? However, we are going to leave the fiber at the front for two reasons. First of all, because as I said, we want a minimal model. So we don't want to get into the complication of having two separate dynamics, one for the cells, the other for the fiber. We want the fiber to move at the velocity of the cells. And the second reason, which is less fundamental and more practical, is because from the point of view of cell density, like how many cells are there per unit surface, actually very few in relative terms are able to pass the fiber. So what you see here is another image that we took in our experiments. On the left, you see the image of the cells. It's like a zooming of the videos that I showed you before. And on the right, we show again the nuclei of the cell. So what you can see is basically that although, of course, there is cell tissue below the fiber, migration goes from top to bottom. So although you see that there are a number of cells that actually crossed the barrier, crossed the fiber, when you quantify the cell density in this part here, actually it happens to be about 1/3 or something like this of the density that cells have at the top. On the other hand, you see that these cells, they are very much spread. So it's reasonable to assume that they are not going to be able to exert as much force as the cells that are here. You can see how look like the day that the sales start, no? Like everyone goes to the department store. So this is basically what you see here. And obviously you can imagine that the cells that are going to be upstream the fiber are going to exert a much stronger force on it than the ones that have already passed the fiber. So that's why, although as I said, we have fibers, we have cells that have passed the fiber. Today, or in this model, we are going to model the fiber as if it was at the front. There is another reason that I will explain later when I will show you the model. Well, and this is the equation. This is basically the model that we have. I think it's the first real equation in the presentation, not bad, and I'm already like half an hour into it. So yeah, so the model that we do is, as I said, as simple as we can do, that incorporates the different effects that we were mentioning. So first of all, the flow is going to be modeled as a 1D compressible, as that of a 1D compressible liquid. Compressible is very clear. I was talking about cell density and how it changes some minutes ago. So yeah, everyone understands that a cell tissue, a cell monolayer, doesn't behave as an incompressible liquid. It's a compressible fluid. So this is the first feature of the model, is 1D and is compressible. The second feature, as I said, is inertia. So we are going to have something that accounts for inertia. And we are going to do it as we do in fluid mechanics. So basically, I have something that is like a density, if it was a fluid, right, times the material derivative of the velocity, or in other words, the convective acceleration. Now, what is this m parameter that I put here? Well, this parameter is basically a measure of the inertia. Of course, as I said, it's not a real inertia, it's polarization. This m is a parameter that characterizes the memory of the cells, how well they are able to keep their state of motion, or in other words, how easily they change their state of motion. So it plays the role of an inertia or a density mathematically, but of course it's not a density. That's why I didn't want to call it rho, I call it m to avoid any confusion. On the other hand, and I'm mentioning this because this is kind of novel in the models that you see formulated in the literature. For the acceleration of the cell, we use the convective acceleration. And we think that this makes sense because inertia has to do not with the tissue, but with the cell. Is the cell the one that remembers its state of motion? So therefore, if I move inside the tissue, I remember the velocity that I had. So therefore, the acceleration that we calculate has to be an acceleration following the fluid particle. Cells today are our fluid particles. So that's why we use the full nonlinear expression for the acceleration. As I said, I'm insisting on this because in many biophysics paper, of course, people try to avoid nonlinearities as much as possible, and therefore the convective term is dropped. And we made the point that we wanted to keep it in our equation. We think it makes all the sense. We have to do the problem numerically anyway, so for us it's not really a problem to have inertia or to have the convective term. Now, the other thing that we have is, let me go to the last one, is a viscous light dissipation. So we can model viscosity, or we can model viscous dissipation in many ways, but we are going to, but the thing is that there are many ways in which cells can dissipate their energy. So out of all the possible ways, we are going to keep only will be some sort of dilatational or extensional viscosity or bulk viscosity, if you want. So basically, when the tissue will stretch or contract, this is where viscosity is going to act. Now, isn't there other kind of viscosity, like some viscosity associated to the friction of the cell monolayer with the substrate? Yes, of course, in principle it would be, but if I keep adding terms to the equation, then it wouldn't be the simplest model possible. So basically, we're accommodating for what we think will be the strongest source of dissipation that we expect. And there is a reason for that, but I mean, we can discuss that in the equation and in the questions later if you want. And finally, as I said, the third element is the elastic fiber. The elastic fiber is going to be modeled as a point force, as a Dirac delta, that acts at the front. And this, well, the intensity, of course, is given by Hooke's law. So the intensity of this force, or the modulus of this force, I should say, is the elasticity constant, K, times the position of the front, okay? One comment. So imagine that we don't want the fiber to be at the front, because we saw that, you know, some cells actually like go past the fiber. Well, that's why we adopted this formulation with this Dirac's delta, because it's a formulation that is going to make easier in the future if we want to say that this XF doesn't have to be necessarily the position of the front. So therefore, this will make our model more or easier, let's say, to extend to other configurations, including the three-dimensional one. So this is basically the equation. And then, of course, this equation needs some boundary conditions to be integrated. So the boundary conditions that we are going to have, well, we have boundary conditions at the front. We have a dynamic boundary condition where basically what we will impose is that the force is that given by or exerted by the fiber. We have a kinematic boundary condition, which basically says that the fiber moves with the position of the velocity, sorry, of the cells at the front. And we have a far field boundary condition. So it's a condition that we will put as far as we can, numerically speaking. Basically, we will say that the velocity of the cells is uniform when there is no external perturbation, such as the fiber. So far away from the fiber, they will keep a uniform velocity field. There is a detail I didn't comment about this dynamic boundary condition. Yeah, the way to do it, the way to obtain an expression for this condition is basically by integrating this equation between a little bit before the fiber and a little bit after the fiber. So in that way, the Dirac's delta disappears. It gives a finite contribution. And then we obtain a boundary condition in terms of U, du dx and xf. That is the one we need to integrate the equation. I'm not going to explain much about the details unless people is interested later in the questions. But of course, we work in dimensionless terms. So we have a dimensionless formulation. And what we do is to scale our variables so that such that the front of the cell monolayer is always at a given at a constant coordinate. In this case, is xi equal to 0. So actually, the problem that we integrate in our numerically is the one that you see here, that is dimensionless. But as I said, I'm not going to enter into this formulation much more because it's a little bit less intuitive than the other one. So, well, this is the problem that we integrate, and this is the result of our numerical integration. So, well, as you see, we are able to reproduce with these three simple ideas, we're able to reproduce qualitatively what we see in the experiments. Namely, at the beginning, we have a flow that is dominated by inertia. So at the beginning, cells just keep moving at a nearly constant velocity. Later on, there is some recoil that we associate with the viscous terms that kick in and start to dissipate the momentum. Although, as I said, it's not really momentum. We will be talking about cell polarization. And then, yeah, the other thing that we see is that, and this is one of the main features that we can recover from the model. The other thing that we see is that softer fibers, softer here means longer, right? Like yellow. These ones, they do, or cells exert a smaller force, which is, as I said, a little bit counterintuitive, but it's a behavior that is observed quite often in cell biology or in cell physics. Well, so these are this is the model, and of course, when I talk to mathematicians, we are very happy with these kind of models because they reproduce inertia, which is something that you don't see very often in literature. So we were very happy with that. But as I said, we work with a group of applied biologists, people who want to be able to obtain more information about what these results may mean from the biological point of view. So what we did was to try to answer a few questions, like, for instance, what is what eventually makes cell stop? And what is what the condition that tells a cell that, enough is enough and we cannot keep pushing forward this fiber? Well, to try to explain that question or to answer that question, what we do is simple order of magnitude analysis, and we focus on the first peak, on the first maximum, because that's precisely the point where cells stop for the first time. So, because at the first maximum, in a damp oscillator, what you have is that the maximum is determined by a balance between inertia and the elasticity. So we are going to do the order of magnitude between those two terms. So basically, I would say that about the time that the cells reach their maximum, the inertia, remember, it's not really inertia, but the term that we model as inertia has to be of the order of the maximum force that the fiber or that they are able to exert, the maximum force that the fiber has measured. And this force, of course, has to be the elasticity of the fiber, K, times the maximum deformation. So that's one idea. The other idea, of course, well, we can talk about the characteristic time, but also we can say that this time at the same time is given by the maximum deformation divided by the initial velocity that the cells have. So then combining the two equations, you end up with this conclusion that says that the maximum displacement that we expect to see for our tissue has to be of the order of this factor here. Now, M and U naught, they are the memory term or inertia term, if you want, and U naught, the initial cell velocity. Now, it doesn't depend on the fiber, of course. These are properties of the cell. It depends on the kind of cell, how do you grow the cells in the first place, how do you start your experiment. But once cells start to move, the properties of the fiber do not affect this U naught nor M. So if we are only going to change the properties of the fiber, we're going to see how cells respond to different fibers, then these parameters, they will be treated as constants. That's on the one side. Now, actually, if I want to obtain force instead of displacement, then I have to multiply times the elasticity, and multiplying times the elasticity, I get this expression for the maximum deformation, where again we have this factor here, which is constant because U naught and M depend on the cells, but not on the fiber. And EI, which is a property of the fiber, is the stiffness of the fiber. Actually, it depends on the fiber, but it's something that we will not change. What we have done in our experiments, as you remember, it was to change the length of the fiber, not to change the kind of fiber. So in fact, this prefactor, this everything but the L, is basically a constant. So it's something that we expect not to change between the different experiments. So therefore, what this order of magnitude estimation teaches us is that we expect the maximum force that the cells are able to exert to decay as the square of the length of the fiber. So this is one of the predictions that our model would be able to make. Now, this is the one concerning the maximum force. And what about the maximum energy that the cells have spent? Well, let's see if the force is actually given by this expression, then we would expect the maximum mechanical work, Wmax, that the cells can exert before they can no longer cope with the fiber and the recoil. It should be of the order of this maximum force times the maximum displacement. With, of course, doing the relevant numbers and doing the algebra, you get that is equal to this expression. But remember, M and U naught, they were properties of the cell tissue. They were not properties of the fiber. So we expect the maximum mechanical work to be the same regardless of the stiffness, in other words, the length of the fiber. And well, as you can see, the agreement with the experiments of these two simple ideas is not that bad, especially taking into account that we are talking about cells, so they do their cell things, they are quite variable. So you see that indeed, within the limited range that we have, that is not that large, but we see that the maximum force decays square of the length for different cells lengths. So this would be 2 millimeter, 3, 4, and 5 millimeter. And although it comes with more error, but I think it's still a reasonable assumption to claim that indeed this constant power curve, this constant product of force times displacement, it reasonably fits the values obtained from the experiment. So we were kind of happy with these results because, as I said, we don't expect to make a quantitative description of the tissue. This rarely happens in this kind of problems in biophysics, but we wanted to understand the main features that our cells are doing when they respond to this elastic object. And we were kind of happy with these results. So yeah, I think we kind of understand well what these guys are doing. And actually this conclusion, this thing of the maximum work, that's super important for biologists because they are able to associate that to the cell production mechanisms that cells have and how some disease can impede the ability of cells to produce energy at the rate that is needed to counteract the object. 2 comments I wanted to make. First of all, as you see, the range of this log-log plot is not very large, especially not in the x-axis, because the length barely doubles. But however, I like to remark that the stiffness, which is the thing that actually enters in the model, it goes as the length to the -4. So in fact, we are in stiffness, we covered a factor of 16 or 20 or something like this. So it's not as small as it seems. So we still think that this is a significant result. The other comment, of course, that I have to make concerns with what most of you will have seen in the first place, which are the points that don't agree the trend. This is what I do always, like always focus on the thing that doesn't work. Well, what happens here is that these are the longest fibers. These are 5mm fibers. And we believe that at 5 millimeters, it's very hard to assume that the cell velocity is a uniform field. In fact, it is not. So they have their fluctuations. It's sort of a turbulent flow, as you probably remember from the video. So we think that basically these fibers don't deflect as if there was a uniform force acting on them. They do more funny things. So that's why these experiments, they are nice, but we expect them to have more error. So well, basically I have told you so far about the things that work nicely in the model. This is something interesting. Well, we have compared already, or the biologists that work with me, they have compared already these curves for different mutants. So they have done it for healthy skin cells, keratinocytes, which are the ones represented with circle. And then in squares, they have a mutant that is called Kindler, and that is the responsible for one of the rare diseases that they study. Now, why are these results important? Because still there is controversy about why these Kindler patients, they have a skin that is not functional. In particular, it's a skin that is not able to close wounds as it should. So, and it's not clear why. So there are people who speculate that is because they cannot exert force, so therefore they cannot push around other tissues, or, I mean, there are different explanations. Well, what our results show here is that they are not particularly worse than healthy cells at exerting forces. In fact, when you measure the velocity field of the tissue, which is something that we also do, you see that these cells, they are a lot more chaotic. So the problem they have is not that these cells are not able to exert forces. The problem they have is that I exert a force in this direction and the other cell exerts in the opposite direction. So at the end of the day, what happens is that there is no coordination, but we don't think, based on these results, that these cells are worse than the healthy ones, or not a lot worse, at least in exerting forces. So you see the kind of analysis that we want to do with the cells. Well, so as I said, I talked about things that work fine in the model, but there are others that don't work as well. For instance, we are not totally satisfied with how our model describes the velocity field. We can measure the velocity, of course, in the cell tissue using techniques like PIV, to basically feed the PIV algorithm with the cell and the cell tissue, and it gets a velocity field for you. And then when you do that, you can compute things that are called chemographs. So a chemograph, for those of you who are not familiar with the concept, I was not before I was starting to work on these things. A chemograph basically is a representation of a one-dimensional field in a spatio-temporal diagram. So in order to obtain these chemographs, these plots, what we do is to measure the vertical velocity on boxes like A, B, and C. We average this vertical velocity on the y direction, like along the narrow direction of the box. And then we do this for different time instants. And we represent this y averaged velocity, which is a function of x, on different columns for different time instants. And this is how we obtain these chemographs. So in color, with this scale that you can see here, we have the velocity of the tissue. White is the position of the fiber. Of course, if you image the fiber near the clamping or at the center or at the tip, you see that the deformation changes. The deformation is larger when you get farther away, of course. And then, well, something that you can see is that at the beginning cells, they sort of have a uniform velocity within the fluctuations that typically you have in these sort of experiments. And then, well, at some point they stop. And they stop not particularly more closer to the fiber. So actually, it looks like they stop at the fiber, but the velocity is a stop already quite deep into the tissue. And this is something that our model doesn't really reproduce well. In fact, if you plot the calculation of the velocity field that is predicted by our equation, what you see is that the fiber will be at psi equal to 0. It will be on the x-axis, no? And what you see is that you expect cells to first of all stop at the fiber, and then the ones that are behind that don't have any perturbation to stop later on. And what we see is not really so. It's like, yeah, actually they stop far away from the from the front first. No, why is that? Well, we believe that is because these cells, they don't migrate if they don't have a reason for that. So when they are sufficiently far away from the wound, it's like, I should not migrate. What for? I have nothing to do. The ones that really want to migrate are those that are close to the front, those close to the wound. So in these kind of experiments, it's very hard to disentangle the two effects, like what happens close to the wound to what happens far away. So say that our model at most could be a reasonable assumption of what occurs close to the wound. Close meaning a distance of the order of the typical distance that viscosity takes to inform that there is a fiber and that cells need to stop. So this is something that our model doesn't do very well. Of course, you can argue that we could always add a term like a Stochastian friction, like a constant v times the velocity u, and this would take into account this behavior. Certainly you do, and there are models that incorporate that in the literature, but we don't want to do that, because if we do that, then we are adding one more parameter. So of course we'll be able to make the model look closer to reality, but in a region where we don't want to, because we are focused on what happens close to the fiber. Okay, so this is basically what I wanted to tell you today. As usual, I think I'm a little bit off with the time. I talk too much. I thought I have very few slides. Well, so basically, the study we did is we want to focus on a real problem, which is how to measure forces on a migrating cell tissue. To do that, we observe the experiments. We put this nice carbon fiber, and we see how cells deflect the fiber. And from the experiments, we observe that there are three main ingredients that we need in our equations. Something like inertia, or an inertia-like term, a viscous-like friction or dissipation, and the elasticity of the object. And when you include these three elements into the model, you actually happen to reproduce quite reasonably well what happens in the tissue. Of course, if you go to the literature, as I said, there are many models, wonderful models with plenty of ideas that describe very well what the tissues do. But still, this is the first model, to my knowledge, that describes the interaction with an object that is not like free cells, let's say, and this is why we are happy with it. And of course, we focus on this. We didn't want to include 7 different terms. We are happy with an equation that has only 3 terms, and one of them is an elastic fiber. So we are very happy with that. And from the more practical point of view, this model already teaches us some important lessons that biologists use to compare different cell strains and to compare different mutants, which is basically we are able to use these ideas to predict the maximum force that the cells are able to exert, or the maximum mechanical work that they can do before stopping. So with this, and a little bit before 3 in the UK, I'm stopping. Of course, I'd like to mention my team, José Luis Jorcano and Juan Las Heras, with whom we developed together the idea for this project, and the 2 women that have been the workforce doing the experiments in the lab, Veronica, the one that is closer to me on the image, and Leticia. And that's it. If you want more information, of course, I'm happy to answer questions. And you have here this paper that we recently published on the study. Thank you very much.
Very good. Thank you very much, Javier. Thank you. Very interesting and very far from beer topic. Yeah.
Indeed.
Any questions, please? I understand that it's 3 and some people should go, but we have time for questions. So I'll stay here, and the seminar is still open. So any questions?
I'm going to change to the other mode so I can see the.
Yeah. Let me. Who is asking? Yeah, Davide, can you? Yeah.
Yes. Hi, Javier. Really nice talk. Thank you.
Thank you very much, David.
I have a really probably stupid question. So when you show the deformation of the fiber due to the force, then you said that you could fit that using the Euler. I don't know how it's called. Euler force, Euler beam. Thank you. Because the deformation is parabolic. I was just wondering, can you get some? Is it possible to get the same parabolic deformation, but assuming a different type of force? In other words, is it unique, this type of deformation, telling you that yes, you only have the Euler beam? Do you see what I mean?
Absolutely. Well, it's one of the problems that we have in this study. Actually, well, not when we did the study, because we saw from the very beginning that it follows very well the Euler's beam. But the problem that you have is that if you want to obtain the force distribution from the displacements, that's an inverse problem. And indeed, it doesn't have a unique solution. That's very easy to see. If I incline my force a little bit and I pull a little bit from the fiber, the force is different, but the deformation will be the same. So yeah, indeed, the solution is not unique. So that's why what we did was to try to go to the simplest case possible and to say, let's assume that all the cells exert the same force at different points of the fiber, which is a questionable assumption, because actually the locally, like the part close to the clamping, is stiffer than the tip. So it's not quite clear that cells will do the uniform force. But the good thing with this is that it gives you one number that then you can use in a 1D model and can use to obtain information of relevant for biology. So, but yeah, absolutely, you're totally right. And indeed, the solution is not unique.
Okay. Well, I mean, I agree with you that you always want to choose the easiest possible model if you can, but I was just curious somehow.
No, you're absolutely right. It's a complicated inverse problem, and the solution is not unique.
Okay. Well, thank you for clarifying that. Thanks.
Thanks to you. Actually, it was a matter of intense debate at the very beginning in the project.
Javier, just his comment. I do not think it's physically relevant to your problem, but kind of similar problem, hydrodynamically similar. If you have a chamber, say, and there is the flow, or you increase pressure, and one of the wall is elastic, say, but not only elastic, but also perforated. So then the first liquid cannot go through, and it's bends the plate. After that, it goes through the holes, so it's appear on another side. So the pressure drops, and it's as you're saying, recoiling back. But after that, it's still goes in the direction of the flow. So that is I'm just tried to understand from a dynamical point of view how the cell could appear behind the fiber, and what the deformation and why it's recoiling back. So from this kind of analogy, I don't want to say that this is the modeling of this phenomenon, but it's kind of something similar, like kind of like a toy, which could explain what's potentially happening. And as to the questions, I have some of them. So why the cells are moving? Is it motion like the same cells, or they just reproduce each other, or produce new cells, and so it's growing material, or
like close to the fiber, we expect to be mostly cell motion. First of all, because they move very fast, and second, because precisely when the velocity is the largest is within the first 24 hours, and within 24 hours, cells barely have time to replicate, because the typical cell division cycle for this kind of human cells or skin cells is about 24 hours. So at the very beginning, they are not able to replicate fast enough. What we have seen in other kind of experiments, and also other people have seen that, not only us, what we have seen is that far away from the front, cells do replicate. So the way it seems to occur in a very simplistic explanation is that those that are at the front, they invest their energy mostly in moving, whereas those that are at the back that don't really have to move much, they are the ones able to replicate to sort of ease the pressure in the tissue, or the tension in the tissue.
So it's kind of interplay between motion and replication, but not on the front mainly, as you said, but inside behind that.
Exactly. Exactly. Yeah. Actually, there is a paradigm in cell biology which is like replicate or migrate, which we don't really see is the case, because we have seen cell replicating close to the front if you give them enough time. But to that point, I mean, there is like a paradigm. Like some people think that they cannot do the two things at the same time.
Yeah, potentially, yes, potentially. And how it's possible that the cell goes behind the fiber? I mean, physically, how it's possible? There goes from the plane in 3 dimensions, and then how is possible?
Yeah, first of all, because the fiber is 7.5 microns in diameter, and these cells are sort of big. They are about 10, 20 microns. So the cell is not really that big. It's like this, no, in human terms, no. So it's not that big. So they are not. It's not they have to climb a wall, no. And on the other hand, what happens is that there is, as you probably saw in these images, there is some accumulation upstream the fiber. I don't know if I guess you can see that, no.
Okay.
So what happens? Cell, although this is a monolayer, because these cells, they like to be grabbing the collagen that is on the ground, so they don't really like to detach from the ground. They try to stay as much as they can, but it doesn't mean that they cannot climb on top of each other, no. And in particular, the fiber is functionalized with collagen, so it's coated with collagen, so they mistake the fiber by part of the ground, no. So fibers are cells are happy climbing on the fiber, and it's easier for them because there is already like some sort of a slope caused by other cells that have accumulated. So
okay. And also one question about the very beginning when you discuss the inertia-like forces or effects. So as to me, it's because it's very slow, right? So it's more looks like that experiments by Kelvin, I believe, when he put the piece of tar on the stair, and in 100 years it moves slowly down the stairs, right? So there is the force. There is no inertia. It's negligible in standard way. But so for me, that polarization you discuss, it's more like force acting, external force acting on the cells, and this is why they are moving. But I don't know physically. And then
it's totally that. Yeah, actually, there are models that they have an equation for cell polarization and another equation for velocity, for instance, and they are modeled with several equations for the different variables. And in these models, they hypothesize things like the velocities, like the gradient of the polarization. So basically, they have like a different polarization field. However, for the reasons that I described before, we tried to avoid it. So we said, well, what about we just put the good old inertia that we have in fluid mechanics, and maybe that is enough to reproduce the equations, no? Or the experiments. No?
Yeah. Any other questions? Yeah. Then one question: what this fiber is for? Why do you need this fiber? What is that?
We want to measure the force. So the fiber is an obstacle that in reality it could be another tissue, for instance, or it could be some sort of bandage that you can put to keep cells compressed close to a wound or something like this. So the fiber is some object whose elasticity is well characterized. So therefore we can measure force out of the formations. And it's something that we can use to force the cells to put some pressure and to see if they're able to do it. No, of course, the next step on the project, if we can continue on this, it would be to make the tissue grow in 3D around the fiber. One of my colleagues, Veronica, she's already trying that. In principle, one of the advantages of this model is that we think that it's not too hard to extend it to 3D.
Yeah, and what you showed that the when two front of cells come to each other, right? They still continue moving, and it looks like as many other humans, I have wounds on my on my hands, and they are still visible, and. It's there is the it's not skin, it's something else, and it's actually a little like hump. It looks like hump. So this is the could be that one.
I think call it scar tissue because it's like an excess of material that grew where there used to be a wound.
Yeah. And finally, if nothing, it's would be not right for me to give English names to such models, but there is a name actually. I could be wrong, so the others could correct me. It's called such models, which are as simple as possible, but they still include the main effect. They are called Persimmonial models.
Oh, Persimmonial model. I like it.
Yeah.
Next time I will talk about this, I will use that term.
Yeah. Because we use it because the objects we study in different situations, in different places on the Earth, they are different, but there are something general between them, and that is what the in the essence of that object, they are making that Persimmonial model, which is definitely in each particular situation is not perfect, right? But it's still able to describe the main issue, main behavior of the Persimmonial.
It's a good
Great.
Yeah.
Great. Yeah. Anything else? There is a okay. It's a message from Paul Hamilton, just saying thank you very much. I have to leave. This is the problem with our seminars. So let's say some people are teaching. So if there are no any other comments, if anybody wants to say something at the end, it's no. Okay. Then I'm closing this. I'm closing this very interesting seminar. Thank you, Javier. So it's
thanks to you for this. And of course, I mean, if anyone has a question or would like to discuss, I mean, you guys have my email, so it will be a pleasure to continue the discussion online.
Yes. Yeah. That's very good. Okay, then I'll stay for a while. So if somebody wants to speak to Javier or between each other, you can stay. But I'll switch my camera and then microphone because I need time, hopefully, to upload the video. No, first download and then upload. So it takes some time. Okay. So bye and Javier, thank you very much indeed.
Thank you very much, Asa. It was a pleasure. See you next time. Hey, Mark.
I have